Hello, my name is Ransom 4, and this presentation is about glycolysis, glycogenesis, and the role of aldolase gene expression in both of these processes, that is, glycolysis and glycogenesis. I will start by looking at an overview of glycolysis. I want to start by looking at an overview of glycolysis. And again, I will be talking about glycolysis, glycogenesis, and the role of aldolase gene expression in glycolysis and glycogenesis. So I want to start by looking at an overview of glycolysis. What is glycolysis? What are some of the things that are specific to glycolysis? Glycolysis is a process by which glucose or a glucose molecule is broken down to form two molecules of pyruvate. Two molecules of pyruvate or two molecules of pyruvic acid. Those two terms can be used interchangeably, pyruvate or pyruvic acid. In glycolysis, equally, a glucose molecule with six carbon is split into two molecules of carbon compounds. And these two carbon compounds, these two molecules of a three carbon compound is called pyruvic acid. Although the ultimate goal of glycolysis is to produce ATP, it is worth mentioning that initially or at the initial stage of glycolysis, ATP is required. So to phosphorylate a sugar, for example, or a glucose molecule, uh, two phosphate from ATP are used. And so this initial step of glycolysis require energy. They require ATP. Again, the ultimate goal of glycolysis is to produce energy. So it's also worth mentioning that the process of glycolysis produces a net gain in ATP. Otherwise, the process will be worthless if at the end we didn't have a net gain of ATP molecules. Glycolysis takes place in the cytosol of the cell. It occurs in two stages, and we usually break them down into the energy requiring stage and the energy releasing stage. Now let's look at the energy requiring phase of glycolysis. In the energy requiring phase, two phosphate molecules are attached to the glucose molecule or to a sugar molecule. The attached phosphate group cause the sugar molecule to be unstable and split into two phosphate bearing molecules. Again, this is a stage where the two ATP molecules are used because those two phosphate groups they come from the two phosphate molecule. Now the two phosphate groups from two ATP molecules are used, and so this stage requires energy. Now, out of the three out of the three carbon compounds, especially for fructose, only one of them is usually glyceride 3-phosphate, and it is the molecule that usually will enter into the energy releasing phase. And looking at the energy releasing stage of glycolysis. In the energy releasing stage, the three carbon sugar via a series of steps are converted to pyruvic acid. So these stages are catalyzed by some enzymes. And then this reaction generates two ATP molecules and one NADH molecule. This cycle of reaction occurs twice, and so we generate four molecules of ATP and two NADH molecules overall. And so, because we have two ATP molecules that are used during the energy releasing stage, during the energy requiring stage, and four ATP molecules that are produced during the energy releasing stage. A complete cycle of glycolysis leads to an overall gain of two ATP molecules. The reaction of glycolysis are all catalyzed by its enzyme. And one very common and vital example of this enzyme is phosphofructokinase. Usually we say it's phosphofructokinase 1 because the phosphate is usually attached to carbon 1, which speeds up or down the formation of the two 3 carbon compounds depending on the energy needs of the body. So that's the energy releasing stage of glycolysis, leading to the formation of four ATP molecules and overall leading to a net gain in two ATP molecules. Glycogenesis. Now, let's look at glycogenesis. Again, in glycolysis, we're talking about breaking down sugar to release energy, to release ATP molecules. Now, in glycogenesis, usually when we have an excess of sugar in the body, uh, excess sugar in the body is not a healthy thing. And so the process of glucogenesis is usually triggered in order to convert or in order to regulate the amount of sugar in the body, right, by converting that into glycogen in the liver, right? So the excess sugar that we have is being converted to glycogen and stored. Now, let's look at an overview of glycogenesis before. Glycogenesis or glucogenesis is a process of glycogen formation. And so glycogen formation stems from, it uses glucose, it uses sugar, right? Glucose is converted to glycogen. When the body contains a high amount of sugar, the body responds by converting the excess sugar to glycogen in the liver. The conversion process is triggered during the rest period after the Cori cycle. After the Cori cycle, this process reduces the blood sugar level in the body and can also be activated by the hormone insulin. So, if you've ever talked about insulin, these are these two pancreatic hormones uh, that work to regulate. You have insulin is one of the two pancreatic hormones, like glucagon. Glucagon is the hormone that will work to increase the blood sugar level in the body when it is low, while insulin is that hormone that will help to bring down the blood sugar level in the body when it is high. So, for sometimes we will often hear diabetic patients saying they want to take some insulin injection. It's because when the body starts failing to produce this hormone, it leads to that disease called diabetes. And so it is one way to remedy the situation is to take in insulin that will help to play the same role as a natural insulin that the body is failing to produce, that is bringing down the blood sugar level. Now, let's look at the steps involved in uh, uh, glycogenesis. So, glycogenesis involves 6 steps. The first one is the phosphorylation of glucose. Sorry. A glucose molecule is phosphorylated into glucose 6 phosphate. The phosphorylation is catalyzed by the enzyme hexokinase. So glucose plus energy gives us glucose 6 phosphate, and the reaction is speed up or catalyzed by the enzyme hexokinase. The second step in the conversion or in glycogenesis is a conversion from glucose 6 phosphate to glucose 1 phosphate. This step is catalyzed by the enzyme phosphoglucometase. Okay, phosphoglucometase is the enzyme that helps in catalyzing glucose 6 phosphate to glucose 1 phosphate as the reaction is being depicted by the picture. Now, the third step in glucogenesis um, is the binding of uridine triphosphate to glucose 1 phosphate. In this step, the glucose phosphate reacts with uridine triphosphate to form uridine diphosphate glucose. So it's an active nucleotide of uridine diphosphate glucose. The reaction is catalyzed by the enzyme UGP glucose, so it's abbreviated uridine diphosphate glucose pyrophosphorylase. So that's the enzyme that catalyzes this binding of uridine triphosphate to glucose 1 phosphate. And so we can easily see that each of these steps is kind of unique in the enzyme that catalyzes it. So the fourth step in glycogenesis is the attachment of uridine diphosphate uh, glucose to glycogen primer. So the OH group of an amino acid tyrosine of glucogen serves as a site for the attachment of the initial glucose molecule. This process usually is kind of a, like it's a very common example of maybe polymerization, as we say in chemistry or in science, whereby you have little molecules that attach themselves on a chain. So uh, when the amino acid of tyrosine serves as that premier, is the process is kind of initiated by an enzyme called glycogen initiator synthase. So the process will always it will always happen in such a way that there is always an open end for an for a glucose molecule to keep attaching onto it, and then that will lead to a molecule, a polymer made up of so many glucose molecules. Now, when we look at the fifth step, here we're talking about glycogen synthesis using glycogen synthase. So here the enzyme synthase transfers a glucose molecule from uridine diphosphate glucose to the non-reducing end of glycogen, thereby forming a 1,4 linkage. So after this fifth step, you now have the glycogen branching, whereby the branching is initiated by the action of an enzyme amylo transglucosidase. Small fragments made up of 5 to 8 glucose residues are transferred to the non-reducing end of glycogen. The process leads to the formation of more non-reducing chain, and the attachment continues until the blood sugar level is balanced. So you always have a non-reducing chain on which a glucose molecule can attach, and so this is going to happen until the body blood sugar level is balanced. All right, so let's check for understanding. At this point, I want you to read the questions, pause the video, and think about it for a second. Let's see if you get it. But if you don't get it, it's okay. We can always watch the video and try to see if we can grasp the concept. So the first question is, where in the cell does glycolysis happen? Glycolysis is the pathway in which dash is oxidized and cleaved to form dash. What are the three functions of glycolysis? During glycolysis, one ATP is consumed to make what molecule? Again, you can pause, check for understanding, and we can always come back to that. Now, let's check. Now, first of all, if we look at the first question, where in the cell does glycolysis happen? Right? Glycolysis happens in the cytosol of every cell in humans. Again, the cytosol is the location in which glycolysis happens. Now, the other one is glycolysis is the pathway in which glucose, right, is oxidized and cleaved to form pyruvate or pyruvic acid. What are the three functions of glycolysis? The first one is ATP generation. So we want to generate energy uh, for body use. The body needs energy. We need energy to carry out all the activities that we do every day. And so the primary reason of glycolysis is to generate energy. The second one is to synthesize a precursor uh, for the synthesis of other molecules like fatty acid, glycerol 3 triphosphate. And the other reason is to synthesize precursors for certain amino acids. Again, ATP generation, synthesizing precursors for the synthesis of fatty acids, glycerol triphosphate, and certain amino acid. Now, during glycolysis, one ATP is consumed to make fructose 1,6 biphosphate. And because this reaction takes place twice, overall, two ATP molecules are consumed. All right. Now, let's look at how this idolase is involved in glycolysis. How is idolase involved in glycolysis? First of all, Idolaters is an enzyme that catalyzes the conversion of the six sugar molecule to three carbon molecules. So remember, during glycolysis, we're talking about a sugar molecule, which is a six carbon compound, being broken down or cleaved into three carbon compound. That step is actually catalyzed by the enzyme called idolase. For example, the conversion of fructose 1,6 diphosphate to glyceride 3 phosphate and dihydroxyacetone phosphate via glycolysis metabolic pathway is catalyzed by idolase. Now, we have a family of idolase. Here we have idolase A, B, and C. Uh, they are all primarily involved in glycolysis. So the A and the C are mostly involved in glycolysis, while the B is primarily involved in both. So idolase C is a key enzyme in the fourth step of glycolysis. It catalyzes the reversible reaction of the phosphorylated six carbon compound sugar to the unstable three carbon compounds that are eventually converted to pyruvic acid. Now, again, we just looked at how idolysis was involved in glycolysis. Now, look at how it is involved in glucogenesis. So, idolase B is the enzyme that expresses itself more in the liver. And so, remember that glycogenesis takes place in the liver because it is in the liver that glucose is converted into glycogen. And so idolase B catalyzes the mechanism of class 1, utilizes a shift-based intermediate for the cleavage of fructose 1,6 phosphate. So it catalyzes the reversible reaction converting fructose 1,6 biphosphate into dihydrogen acetone and glyceride 3 phosphate. And that takes place in the liver. The enzyme work on a six reversible reaction in glycolysis and glycogenesis. Aldolase B is the enzyme that catalyzes glucogenesis. So aldolase B is plays a very big role in glycogenesis in the liver. Now let's check for understanding again with some few questions. Again, because these are multiple choice, the questions look a little bit many, but they're actually not many questions. We only have four questions. So glucogenesis differ from glycolysis because that's the first one. We have a series of options. The irreversible step of glycolysis are bypassed. B. Different enzymes are involved. C. Biotin is required for glycogenesis and not for glycolysis. D. All of the above. E. None of the above. For the second one, the three enzymes that catalyze irreversible reaction in glycolytic pathways are A. Hexokinase, phosphofructokinase 1, and pyruvate kinase. B. Aldolase, phosphoglycerate mutase, and pyruvate kinase. Hexokinase, phosphoglycerate kinase, and pyruvate kinase. That's D. Aldolase, phosphoglycerate mutase, and enolase. E. Phosphofructokinase 1, phosphoglycerate kinase, and pyruvate kinase. Now, question number 3. All are important reasons to phosphorylate glucose in the first step of glycolysis, except while question number 4 is in glycolysis, a phosphorylation step is catalyzed by. So again, it's important to read the question, try to see if we understand them, and then take some time. You can pause the video, a couple of seconds, come back to it and check the options. All right. So the right option for A is for question number 1 is all of the above. So in glucogenesis, it is distinct from glycolysis because of all of those reasons. The irreversible step of glycolysis are bypassed. Different enzymes are involved. Biotin is required for glucogenesis, but it's not required for glycolysis. And for question number 2, the three enzymes that catalyze the irreversible reaction in glycolytic pathways are hexokinase, phosphofructokinase, and pyruvate kinase. For question number 3, all are important reasons to phosphorylate glucose in the first step of glycolysis, except the large positive free energy is important in getting the pathway started. Now, question number 4. In glycolysis, a phosphorylation step is catalyzed by phosphofructokinase 1. All right, let's move on. Now, let's look at the different isoforms of idolase and their different expression pattern. So, the idolase is a key enzyme involved in glucogenesis and glycolysis. And the different forms are idolase A, B, and C. The different forms of idolase differ in the way and location in which they express themselves. Idolase B is preferentially expressed in the liver. Again, as we said earlier, I was talking about idolase B actively involved in glucogenesis, and glucogenesis takes place in the liver. So that is where idolase B will preferentially express itself. For idolase A, it will preferentially express itself in the muscles and erythrocytes, while idolase C is preferentially expressed in the brain, to be precise, in the cerebellum of the brain. So the early expression patterns of A, B, and C. So uh, this study that was looking at the early expressions of idolase A, B, and C in the early development of the embryo. And so the expression pattern of all three idolase isoenzyme genes in different tissues and early embryogenesis. In the study, the grass carp was selected as a model due to its capacity to metabolize glucose. According to the cDNA sequence of idolase B, C, and ABC genes, it was observed that it consists of 364, 364, and 363 amino acids respectively. So idolase A has 364 amino acids, B 364, while idolase C has 363 amino acids. So the expression pattern showed that A was high in the muscle, idolase B was high in the liver, and idolase C was highest in the brain. So all these patterns were actually studied and observed. Now I want to look at the regulation of idolase B by transcription factors NHF1, NHF3, C slash EPB, and GPB. So these transcription factors, it's important to note that they all play the same role in the regulation of idolase B. They all play the same role. All the transcription factors do not actually regulate idolase B expression, but they influence the feedback loop that will allow for the expression of idolase B. A research conducted by Kahn and Pichat revealed that DPB and HNF3 are strong transactivator of idolase B promoter, while C slash EBP and VHNF are weak activators. In hepatocytes, the strong activator of idolase B is C slash EBP, while GPP is inactive. NHF3 as a very strong activator, behaves like a very strong activator of idolase B and will antagonize the trans activator HNF1. The negative effect of HNF3 of the trans activation potential of GBP are unique characteristics of idolase B. So this is a brief summary of how idolase B is being regulated, or is being, yes, it's being regulated by the factors HNF1, HNF3, C slash EBP, and GPB. All right. Again, thank you so much for your kind attention, and I want to express my deep gratitude to the following sources, without which this presentation wouldn't have been given birth to. And so these are the following sources that were used to prepare this presentation, and I want to give them the credit they deserve. And thank you for your kind attention. Thank you for following up or following through the presentation. You made it to the end, and I want to appreciate that. Thank you, and let me know if you have any question.